All right, well, good afternoon, everybody. Happy Monday, and welcome to the last week of the semester. Yes. So to celebrate, the final lecture will be a guest lecture from Professor Greg Underhill from the Bioengineering Department. So welcome, Professor Underhill, and thanks so much for sharing your work with us.
Thanks.
So again, this is a chance for you to see some of the latest and greatest. Some of the new applications of microarrays and applications in stem cells. Very exciting things that I think that you'll enjoy learning about. So without any further delay, thanks for coming, and look forward to hearing your talk.
Thanks. All right, well, hello everyone. Semester's almost over. Good luck on the quiz I think you guys have on Wednesday. I recognize a few faces that I had in classes last year, 476. So definitely interrupt me throughout. We can make this interactive, kind of go faster or slower as you guys want. But so essentially today I want to talk about a couple stories from my research lab's interests that are related to biosensors in a couple ways. First off, one of our main focuses is trying to understand how cells essentially integrate signals in their environment. So cells are sitting in a complex environment. I'll go through more on that. And then essentially they integrate all those signals to essentially leads to some kind of cell fate. Stem cell differentiation I'll talk about today, some cellular function. And so essentially it's the other side of the coin of essentially what I think you guys have been talking about this semester of sensing what the cell does. So we want to control the environment of the cells and then integrate that with a variety of sensing technologies. And then secondly, I think you guys have talked about this idea of increasing throughput, increasing content of your measurements. And so we focus on that a lot as well, focused on stem cell differentiation. So those are the themes that I'm going to be talking about mainly today. But I want to give just very briefly some perspective, some background, and some of the terminology I'll be discussing. So I'll be discussing essentially stem cell fate and function. And so it's becoming increasingly clear that for both adult stem cells, so there are adult stem cells in your various tissues, as well as embryonic stem cells that are in the embryo that can become all the different tissues. Both of these different types of stem cells, as I mentioned, essentially integrate signals that are in their environment. And those signals interact with various intrinsic programs. So this is the genetics of the cell, the epigenetic changes in the cell. And these things are integrated together to essentially lead to some cell fate. So these microenvironmental signals we'll talk a lot about today range from soluble factors, so growth factors, things that are secreted, and then in the laboratory environment, things that are in the media of the cells, for example, how cells interact with each other directly, the cell and extracellular matrix interactions. So today I'll talk a lot about ECM, or extracellular matrix. So those are the proteins in your tissues that provide mechanical stability. Cells adhere to these proteins. They also provide a lot of signaling cues to essentially tell the cell what to become or where to migrate, for example. And then there are various mechanical forces. So this is a very complex local environment that cells integrate. And so how can we begin to explore that? And so a little bit more background on the particular cell system I'll be talking about today is essentially this differentiation of liver and pancreas. So an embryonic stem cell is pluripotent, so it can become all the cells of your body. And so essentially the first fate choice that cell sees, so in the embryo, that ES cell can become essentially either ectoderm, which then becomes neural lineage or skin. It can become mesoderm, which leads to heart, blood, or muscle. But it also can become what's called endoderm, which leads to liver, pancreas, lung, and several other digestive tissues. So this is the fate decision that we focus on primarily in my research group. So once an embryonic stem cell becomes what is called endoderm, then it has this choice of becoming liver and pancreas, and I'll tell you a little bit more about that. And these two tissues are extremely important for translational tissue engineering applications. There's a shortage of livers and for patients that are going through acute liver failure, and various diabetes applications, there's a shortage of cells within the pancreas that could potentially make insulin. So there's an interest from a translational perspective, and in a second I'll talk to you about some of the subtleties of the embryo development itself. And so that's kind of illustrated here. So what happens is once the cells have become endoderm, then those cells are growing in the embryo. And what's very striking is essentially in this developing embryo here, you have a region, in a very specific region of the endoderm, which becomes liver. And then right next to it, that different cells become pancreas. So what controls this specification? Why cells in one area and just hundreds of microns over become pancreas versus liver? So what controls that? So there are various elegant kind of studies within developing mouse embryos that have identified a few factors, and mostly these are secreted factors. So these are soluble factors. And it's been seen that essentially the developing cardiac mesoderm right here plays a big role. Essentially, cells that are nearby this cardiac mesoderm receive some signals that cause it to become liver, and cells that are slightly further away don't receive these signals from cardiac mesoderm and tend to differentiate more towards pancreas. So essentially, these localized cell-cell interactions play a big role in determining if a cell decides to go one way or the other. But besides these few cues that have been identified, essentially not much else is known. How does the liver expand? There's been some thinking that the blood vessels play a big role in that. But again, as I mentioned, there's kind of this striking phenotype where there's a very clear border of cells become liver on one side and pancreas on the other side. So how could this possibly happen? And so one thing that we thought is maybe extracellular matrix, things like collagen, fibronectin, things that are deposited outside the cell might actually set up a network which helps define do stem cells become one thing or the other. And that has essentially not been explored up to this point for these types of stem cells. So what we want to do is understand how these cells integrate these signals, such as ECM, and how that drives this one way or the other. And so what I'm going to mainly talk about today is an idea of using a cell microarray approach. So I think you guys, Brian mentioned that you talked about DNA and protein microarrays in class. And so this is essentially kind of building on that technology. And so what we have here is essentially you can make combinatorial mixtures of various proteins in a 384 well plate. And so that's just illustrated here. And then you use a robotic spotter, which was originally developed for making DNA microarrays. And then you're able to pick up these solutions from a 384 well plate and then print it onto a surface here. And the surface that we utilize is a dehydrated polyacrylamide. So the same polymer that's used for STS page gels, if you guys have ever done Western blots, and often used in several different biomaterial applications. And so the nice feature of this polyacrylamide is it's dehydrated, and then you print your ECM mixtures on top. And then once you hydrate it, so once you add a buffer to it later, then the polyacrylamide is very hydrophilic. And so it's referred to as a non-fouling surface. So it essentially prevents other proteins or cells from attaching. So then essentially what you have here is a surface which the cells or any other proteins can only interact with the domains that you print specifically. So now you have these various islands here where you can have proteins that you individually define, and then as you add cells on top, they can stick to those proteins and maybe adhere if they have the right receptors, but they can't stick anywhere else on the gel because it's very hydrophilic. So then essentially what you can do is you seed cells in there, the cells attach if they have the right receptors, and then you can analyze them in culture. And so this is essentially a cell microarray. And again, for the model system we're utilizing here is this definitive endoderm. So we have, these are mouse embryonic stem cells. So we have those stem cells, we differentiate them a little bit, so they're at this endoderm stage. They have a fluorescent reporter, so we can use flow cytometry to sort out cells to make sure we have a very pure population going into our experiment. And then we add these endoderm cells on top of our arrays that we define here. So, and these are arrays of combinations of ECM, which I'll go through more in a second. And then we can perform these combinatorial experiments where now we've highly defined the environment locally of what the cells experience. So this is one island of an ECM array, and these are GFP positive endoderm cells. So you can see these cells have stuck down to the ECMs that we print. And this is just a couple hours after the initial printing. So the cells are, or initial seeding of the cells. So the cells are just kind of starting to spread out and attach. So what are the specific experiments? Hopefully you can kind of see. But what we wanted to do here was essentially explore a very large space of ECM proteins that we were using. So we looked essentially to try to define what are all the ECM proteins that were available commercially. When we initially did this, there were these 38 ECM proteins that we could find that were available commercially. And the idea from this would be then we would do all these different combinations of these ECM proteins together and print them onto our ECM microarray. And so in order to put this on one slide, what we focused on was all the pairwise combinations of these ECM proteins. So essentially two-factor combinations. So all the two-factor combinations as well as the single-factor controls gives us 764 unique combinations of ECM that we can mix in at 384 well plates. And then the idea is that then we can print those unique combinations onto our, this is a normal microscope slide. And we can also do replicates of each of these combinations. And so what you can see here is that one of these ECM arrays where we've stained with antibodies for two different components. One is brevican, which is stained in red, and the other is chondroitin sulfate, which is a glycosaminoglycan, with basically a sugar molecule, which is stained in green. And so essentially what you can see here is all the combinations that have brevican are labeled in red, and all the combinations that have chondroitin sulfate are labeled in green. And then there's one combination on the array which is actually labeled with both, which is both green and red overlaid yellow here. So these are all these unique combinations of ECM that we can do on this single microscope slide array. So including controls, we essentially have 4,000 individual spots that the cells can stick to. So this is a type of high throughput cell microarray. This is an overlay image of essentially independently labeled measurements of each of these ECMs. So we confirm the presence of each of these different ECMs and then overlaid it in this single image here. So what can you do with it then, right? So we have these endoderm cells, these partially differentiated ES cells, and then we add it on top of the array. And then essentially what you can try to identify which are the proteins that the cells interact with the best. So what we essentially did here was just stain for nuclei and measure the fluorescence. So we're looking to see what are the combination cells stick to, what can they adhere to. And so this is a endoderm adhesion assay, essentially. And so that's what, if you guys start to study stem cells or look into the literature at all, one thing that's really increasingly difficult with stem cells is that as you differentiate them, partly they become more difficult to handle. You can't passage them. It's very difficult to control their differentiation in culture. And so that's what was notoriously known about endoderm. Once you started the process, you can never really trypsinize them or detach them because it wasn't clear what they would stick to. So this was very informative just initially, and this is the type of one way that we analyze this data. Essentially, we can generate this heat map here, where these are the two-factor combinations of ECM illustrated here. And then we scale this to the average, so the mean for the array is set to 0, which is black. And so any ECM combination that supports adhesion much better than that is red, and any one that's much less than that average is green. So essentially from this we can identify what is our endoderm adhesion profile, which is, as I mentioned, was not really clear initially. And so that's the first pass. But then as you start to think about how do cells differentiate, what happens to the cells as they differentiate, you can kind of do some of these more interesting, potentially, next experiments. So the idea here was that, as I mentioned from embryo studies, people have identified some of these soluble factors which are secreted by mesoderm, which cause endoderm to become liver. So what you can do is you add cells onto your array. They initially attach, and then you essentially change the media of the cells for the entire array and add soluble factors that would induce the cells to go one way or the other. And then, so essentially what you do is now you have these 4,000 spots, cells under different conditions, all in one slide. You change the media, try to induce them to liver, and then you see what happens on each of the individual spots. And what was essentially very striking was the minute you induce differentiation, essentially, within minutes, hours, and then definitely within days, as you can illustrate, what's illustrated here, is the adhesion profile essentially drastically changes. So this is a heat map, again, of ECM combinations, and these are under different culture conditions. So I just want to point out, so there are a lot of ECMs that the cells initially attach to, but then once you induce differentiation, the cells come off of those spots, essentially. And so there are a couple of reasons why that would happen. But, and I'll talk about that more in a second. But essentially what you can use this then is to identify the adhesion profile of cells at different stages and then how that evolves across when you have this differentiation process.
Question.
Yeah.
So you just measure adhesion or loss of adhesion by disappearing of the cells, so if they come off, then the fluorescence is gone, and then you...
Right. For these studies, it was just by the presence of the cells. So we stained for nuclei and counted the nuclei. So these are cells that are detaching from the surface, and there are a couple mechanisms for that. But you can just do this in high throughput by scanning under the microscope the entire slide and then quantifying the number of cells or intensity of nuclei for each of the individual spots. Yeah,
These are each different slides.
Yeah, these are different slides.
It's not the same slide in each of these different time points?
These are individual slides, different slides, yeah. You could do time-lapse imaging to try to track one slide over time, but we had a separate cohort of multiple slides at day 1 and then a separate one at day 4. So, yeah, Brian.
Okay, so do you use a fluorescence microscope to measure all these, or is it some kind of microarray scanner?
Yeah, so we've done both. We have done lower resolution microarray scanners, but we wanted to try to move more towards single cell quality imaging. So then what we actually settled on was using a fluorescence microscope and then just setting it up to scan across the entire slide, taking multiple images, and then stitch it together.
So then by the time you measure these, are the cells, are they fixed or are they still alive?
For these, they're fixed. So they were a separate batch of slides, which we fixed the cells and then stained the nuclei or other markers I'll show in a second. And then, so then there's not a rush. It usually takes, at 4x, it would take about 2 hours to scan across the entire slide. So that's how we did that. And then, again, this data is averaged over multiple slides taken at each of those time points.
Question?
Yes.
Sure. So you were saying like the cells within the circle, it's like they've been falling off by the time. But look downside.
Yeah.
Just like, I don't understand, like there's no cells originally, but there are cells later. How's that?
Right, yeah, so it's important to point out that, again, this is all normalized to the average. So it's essentially relative to the mean at that given condition, so we normalize it. So essentially, these conditions here, they were very relatively low compared to the average, but because there are less conditions that are binding at a later time point, these that preferentially bound would increase. And then there's also the effect that there could be cell proliferation during that time. So that's sort of taken into account here too. So it's adhesion and survival and proliferation kind of all essentially connected together in one measurement. So that, I'm not sure about these specific combinations, for instance, that you're pointing out. But it's essentially those cells are relative to the mean are more predominant, and there's a potential that the cells were proliferating as well under those conditions. So what was happening here? And also, I'd like to point out that if we looked at hepatic differentiation or liver differentiation versus pancreas differentiation that we try to induce, the adhesion profiles are also different. Yeah?
Sorry, going back to the earlier point, is it possible that the cells that you saw that had a decrease in adhesion were actually just proliferating, so they detached in order to divide, and then in that process got moved over to cirrhosis and stuff?
I think it's less likely. Most cells will maintain some attachment during that process, but I can't totally rule that out. That there would be I didn't talk about the spacing. So for these arrays, the diameter of one island is 150 microns, and then there's 450 micron center to center spacing. So they're not right next to each other, so they would have to float around a little bit. We can't totally rule that out though, but I think it's less likely to be the situation. So what are some of the other mechanisms that could be causing this? One thing is that the cells essentially could be changing their profile of adhesion receptors. Like maybe they bind to different ECM now that they've been induced, and that would cause them to detach. Another aspect could be that the cells could be actually triggered to go into apoptosis if they're not on the right ECM. And I'm not going to go through all the details here, but suffice it to say, I think it's a little bit of a mixture of both, and that there are conditions in which we did this over multiple days, kind of tracking again, individual cohorts of slides that were staggered for multiple days. And what you can do is you can stain for cleaved caspase 3, which is part of the induction of programmed cell death in these cells. And what we did find was different ECMs essentially had different levels of cleaved caspase 3. So there were conditions where it looked like cells were inducing this programmed cell death, and then at a later time point there were less cells on those islands. So essentially, if the cell is not on the right ECM and you induce liver, for instance, those cells can induce apoptosis and essentially just die instead of differentiating. So we're continuing to look into this mechanism, but at least it suggests that this is part of the story, so that if cells aren't on the right ECM when they're differentiating, they actually induce a programmed cell death. So that's just essentially cell numbers. But you can also look to see what is the quality of the differentiation towards the various lineages. So you can also, using antibody-based fluorescence staining, you can look for markers that are indicative of your differentiation one way or the other. And so this was data where we stained for a protein called alpha-fetoprotein, which is expressed by liver. So it's indicative of liver differentiation. And then we generated another heat map where this is essentially the amount of liver differentiation on each of the ECM combinations. And this is a dot plot. This is AFP intensity versus, again, nuclei intensity, which is a measure of cell number. And then we could identify several ECM combinations, these filled diamonds here, which give a statistically significant expression of AFP and cell numbers. So essentially, these are our hit combinations that seem to promote more liver differentiation. And I've listed a few of those here, and I won't go into all the details of that. But we can also do this again for pancreas differentiation. It gives a slightly different profile. And what we found was interesting was that there were some combinations that were different, ones that supported pancreas versus liver, or liver versus pancreas. But then I've also highlighted a few here, which laminin and superfibronectin, or fibronectin and morosin. Morosin is laminin alpha-2, so it's a form of laminin. So essentially both of these are laminins and fibronectins combined. And those seem to actually support both liver and pancreas. So they seem to support both directions, which I think was interesting. And I'll come back to a way that we use that in a second. So this is our way of trying to identify which ECMs are better for differentiation towards one lineage or another. And essentially one thing I want to point out, so what we found in doing many of these types of studies is essentially combinations matter. That's the take-home message here, is that because cells experience these very complex combinations of proteins and signals, that they you actually, combinations of factors lead to synergistic or antagonistic effects that you wouldn't be able to really anticipate or expect from doing the single factor experiments. And so there are situations where, for example, illustrated here, fibronectin is very good at maintaining the cell attachment. Morosin is really not good at all at cell attachment or differentiation. But if you add some rosin to your fibronectin combination, now you have an increase in the differentiation. So rosin seems to provide this supportive role. And similar with various collagen combinations, where there's synergistic effects of having a very unique combination that you may have not anticipated from the single factor experiments. So again, driving home this idea of if you can increase throughput, then one of the main benefits you get is this ability to do combinations. You're not just doing serially one factor after another, but you can test lots of different things together. And so that's sort of the main take-home message from this. Yeah. Ryan.
So I guess there's a thought that in early embryo development that early progenitor stem cells are attached to a common matrix, but there may be a gradient in that matrix that I guess might result in a breakpoint that results in things on the left going one way and things on the right going the other.
Yeah, I think that's part of the idea, that there may be some common matrix, or there could be one factor that's maybe kind of added to that combination that sets that combination apart from something else. And so we're starting to try to do some embryo studies to try to do stainings to look to see if that's actually the case. But I think that is the idea. Either it's a gradient of combinations of factors, or maybe a very localized ECM in one area and another may work together with these soluble factors to set that up. But right now it's kind of an open question, and that's why we want to do these types of studies. But, and then I just wanted to point out that you can start to look at what the mechanism is. So I won't go into all the details, but one of the soluble factors that we were working with was BMP. And essentially, BMP is known to induce a specific signaling pathway inside cells called the SMAD pathway, SMAD158 pathway. So what we were interested in here is you can actually stain for phosphorylated SMADs. So that means if that pathway is activated, then those proteins become phosphorylated. And so you can stain for that inside cells as a measure of if that pathway was activated or not. And so what we found was that actually, so if you add the hepatic condition, which has BMP, and then they're on different ECMs, you see differential induction of these SMADs. So what this suggests essentially is that if the cells are on the right ECM, then they can better respond to the soluble factors that are around the cell. So it's essentially setting the cell up to respond better to the other factors that are secreted. And so this could be part of that mechanism. So if cells are on one ECM versus another, then they would actually respond to the soluble gradients in a different way. Yes?
Are you controlling the ECM in any way, or there's properties? Is that something that you did to study?
Like the mechanical properties or some, not in this study. We were just controlling, essentially combining it with other factors. But there are aspects to that could play a role, like if it was on a different material with the different mechanical properties. And one thing we didn't look at much was dose, actually. So if you had an increased concentration of that, we kept the total ECM concentration constant here. But there are aspects to that could play a role as well. But here is more of an on or off thing. So essentially, this is where we started to explore the mechanism of what was happening. And kind of last part of this story before I talk about kind of one other aspect that we're working on in the lab is that I mentioned how one of the ECM combinations, fibronectin and rosin, seemed to be good for both liver and pancreas. And so what we did was then essentially use that as our base substrate, illustrated here, and then combine that with a library of drugs. So we were, in addition to kind of identifying the ECM component, we wanted to further explore potential drugs or soluble factors that would allow you to further refine the system. So we picked a library of drugs. These were 800 different small molecule inhibitors that go into the cytoplasm of the cells and block various pathways or induce various pathways. And then we performed a kind of combined screen here, looking again at liver versus pancreas. And I won't go through all the details here, but essentially we were able to identify a few drugs that essentially could replace this part of the differentiation cascade. So if you want to get liver cells down here, what you usually do is differentiate cells in one media for a few days, then change that to another media for a few days, and then change that to another third media for several days. And so what we were able to do from this screen is identify some drugs that could replace the BMP FGF here in this kind of intermediate step in inducing the liver. And this essentially helps you as if you could develop these small molecules. One, they're a lot cheaper than these recombinant proteins, and so they can essentially be translated better to stem cell differentiation protocols. And secondly, because their targets are known, you're able to further explore the kind of the internal mechanism in cells and what's leading to this process. You're able to refine essentially this cellular process. And we did this with mouse stem cells as well as human stem cells, kind of moving towards more of a translatable kind of process. So that's sort of kind of where things are as far as utilizing the cell microarray. This is a two-dimensional cell microarray. And I just wanted to point out a couple things, so kind of ongoing directions, as Brian alluded to. And what I mentioned is that we, in collaboration with another lab at UPenn, we're interested in trying to stain essentially embryos and try to define if there are embryonic ECM domains and how that relates to some of our data that we identified in an in vitro system. We're trying to explore what are these pathways that are underlying this system. So why does ECM and the soluble factor work together to cause the stem cell differentiation? We're also interested in further increasing our throughput. So right now we had a single slide where you had lots of different combinations printed on a surface. But if you wanted to do more combinatorial experiments with essentially media components or soluble factors, one thing we explored is essentially printing these domains into a multi-well plate format, like a 96-well plate. And so we've done that some where we can print 36 different spots into a single well of a 96-well plate. And then these are mouse ES cells that have a red fluorescent reporter that essentially are growing on these individual domains in the 96-well plate. And so now each of these wells could have a different media or different drug, and then you can do those experiments in combination with the various proteins that you print down onto the surface. So this is one way that we could increase our combinatorial throughput. And I think some of the questions alluded to this idea of gradients of factors, and then also how do cell-cells interactions kind of play a role here. And so these are really challenging aspects which we're trying to build on now, not just doing on and off, but actually looking at dosage combinations. And kind of secondly, the experiments that I showed up to this point were essentially just one pure cell type that we put down onto a surface. But we've also explored this idea of integrating multiple cell types. So if you have endoderm as well as mesoderm that you add together onto a combinatorial array, then you may see differential effects. So these are sort of these next challenges of increasing the complexity, the ability to explore these different combinations of factors. And overall, we'd like to integrate this with some more high throughput and high content measurements, not just fluorescence, potentially some of the sensing technologies you guys talked about in this class. How can you essentially get more bang for your buck, right? Like how can you measure more things from this island? What's happening to the cells? What's going on in this array format? So that's something where we have a lot of interest in. And then lastly, in the last little bit of class, I want to talk about a different system, which is slightly different, but again, another focus on kind of increasing throughput. And that's a way to essentially explore how cells behave in 3D. So in the body, in normal differentiation, cells are not on these flat two-dimensional substrates. They're within a complex three-dimensional environment. And so this kind of fits into this overall goal of tissue engineering, which is sort of illustrated here, this idea that you can culture cells within three-dimensional constructs, you can explore their three-dimensional behavior in the lab, or you can potentially even implant them as an engineered cell-based therapy. We're stalled here. Okay, and so when you're trying to explore these types of studies, there are lots of different design parameters. We talk about this in tissue engineering class a lot. What are the things that are going into your system? You could have various cells that go into your system, kind of countless modifications to your three-dimensional scaffold that you can imagine. The materials, their mechanical properties, how you fabricate the scaffold, and then all these combinations of cues that I talked about that you could potentially incorporate in. So how could you essentially increase your experimental kind of space? How could you test all these different things? So one idea that we had was we wanted to increase throughput. So essentially, if you, instead of just doing one large bulk scaffold, if you tried to miniaturize this down to the microscale, so then you can essentially look at hundreds or thousands of these scaffolds. And if you then have a population of these, then potentially you can kind of analyze multiple combinations in parallel, and it would also provide flexibility for lots of different types of assays. So essentially this idea here was we were going to miniaturize our three-dimensional constructs that have cells down to the microscale, and then it's very analogous to have flow cytometry or flow-based analysis of single cells, and then you could analyze a population of these miniaturized 3D constructs. And so there are various ways, various materials that you can use for this. The examples I want to show today are we were using polyethylene glycol-based hydrogels or PEG-based hydrogels. There are various reasons why those are very commonly used in tissue engineering and three-dimensional models illustrated here. They're biocompatible. They have very high water content, so they have very tissue-like mechanical properties, and they're very tunable. So it's essentially a synthetic blank slate that you can modify in any way that you want. And then you can also polymerize them in the presence of cells. So if you have cells inside the pre-polymer solution, and then you expose the cells to UV light and a photoinitiator, then this will cross-link the hydrogel, and the cells will be embedded inside the hydrogel. And there are various ways where you could try to miniaturize this down using microfluidic droplets or molding. One way that we used in some of our initial experiments was essentially photopatterning. So essentially, if you have your pre-polymer solution with cells and then you expose it to UV through a mask, illustrated here, then you essentially will have these microscale domains that polymerize, which is illustrated here. And then for these experiments, we could essentially just release those from the surface, and then you have hundreds or thousands of these miniaturized domains in suspension. So then how could you analyze this? So what we did to do this was essentially use what is called a large particle cytometer. So I'm not sure if you guys talked about flow cytometry. You didn't. So essentially, this is the equivalent to flow cytometry, but it's just for a larger diameter. So this can analyze up to 500 micron size particles. And it actually was originally developed for sorting C. elegans or worm embryos. So you can actually sort embryos using this. You can also sort larger things like pancreatic islets, so regions of the pancreas where the beta cells are, or large aggregates of multiple cells. And so there are a few companies that sell this type of instrument. So it's essentially equivalent to a flow cytometer, but for larger things. And so in this way, we can analyze the entire these are around 300 micron PEG hydrogels that have cells embedded inside, and we can analyze that in flow. And so this is an example of some staining where we just did viability staining. It's where you can look at survival of cells within these PEG hydrogels. So that's illustrated here. And this is an example of a histogram that you can obtain from this large particle cytometer. So this is under the green channel, so we're looking at calcium AM viability. And then now you have essentially a population of these microgels that have cells. And one thing I want to illustrate here is essentially you're increasing the N value that you can obtain for your study. Instead of just having one bulk construct where you measure some bulk measurement, now you can have hundreds or thousands of measurements that you're taking of a population. And you can do power analysis to see how that essentially enables you to improve your statistical assessment. And so let's say, for example, you wanted to perform an experiment where you treated these cells with some drug and then you wanted to look at toxicity. And so the TC50 is defined as where the viability is 50% of control. And so if you do power analysis where you could essentially, you would need 30 of these microtissues to identify a twofold difference with high power and high statistical significance. And in this experiment here, for example, we had 338 of these microtissues. So at high power, you could detect as low as a 1.24-fold difference in viability. So this idea of increasing your N value, essentially, to improve your statistics of your analysis. But also, because these microtissues are in suspension, they give you some flexibility performing various assays. And one thing we wanted to do was this idea that maybe you could actually implant them into an in vivo system and then analyze them after you extract them. So I'll go through a little bit of the setup for this experiment. Essentially, we were using a liver progenitor line. And what we did was we added two viruses, adenoviruses, that we used to infect the cells with various reporters. One reporter is a red reporter, RFP, and this is constitutive, so it's always on. So if the cell is alive, it'll express RFP. And then another was a GFP reporter that was using a TET-off system. So essentially the drug tetracycline or doxycycline will cause, if the cells are treated with that, that will turn the GFP off. If it's not in the presence of that drug, the GFP will be on. So this is, it's a very commonly utilized system. There's both a TET-on and TET-off systems. So essentially the idea that we pursued here was we made this population of microtissues that contain these cells with these reporters. We then embedded the microtissues inside a dialysis tubing, so just a molecular weight cutoff tubing, and then implanted that tubing into the intraperitoneal space of a mouse. Kind of stitched the mouse back up, and then let the mouse sit for 2 days, 48 hours. And what you can do is then you can provide the mouse actually this drug, doxycycline, in the drinking water. It will be absorbed and go through around in the mouse's circulation. And then we were interested if the microtissues were viable, if we harvested them again from the tubing after 48 hours, and if they received the treatment from the systemic drug that we're providing in the drinking water. And that's illustrated here. So the RFP remains the same, plus or minus doxy, which is what we would expect. And then you can see the GFP signal here is downregulated in the mice that receive doxycycline in the drinking water compared to the control mice. So in fact, the microtissues are inside the intraperitoneal space, and they are receiving the drug that's circulating around in the mouse. And this is about a 6.6-fold reduction. So this is an initial proof of concept that we could take these high throughput microtissues and they would receive systemic stimulus within the circulation of the mouse. And then a couple more things that we showed with this system is this idea that, very similar to flow-activated cell sorting, you can sort microtissues in this system. So these hundreds of micron-sized particles. And so we were interested if you could sort out microtissues that differentiated one way or another. So again, this is a reporter for endoderm that we looked at here. I won't go through all the details, but essentially under these conditions, a very small percentage of the microtissues become endoderm, and we were able to sort those out of the system. So a very small percentage become what we want. We were able to sort those out, and then we could confirm that. So once you had sorted them, we can lyse them, get protein or RNA, and confirm their expression, and that's illustrated here. So this is quantitative PCR showing that in fact they are kind of what we thought they were, that those were the ones that became more endoderm. And lastly, I wanted to talk about this idea of multiplexing. So if we really want to increase throughput, then a nice feature is if you can actually perform multiple experiments in parallel within your batch system. And you can do this in a couple different ways. One way we did it was essentially you can add what we call pre-assembly codes. So you could add fluorescent particles to your system that would identify one set of microtissues from another, and then even if they were mixed together, you can measure their fluorescence, and they would essentially encode your multiple conditions that you could analyze in parallel. So this is a pre-assembly code illustrated here. So you could have potentially microtissues that are green, ones that are red, ones that have both green and red, or ones that don't have either of those. And you can identify them separately on the dot plot illustrated here, and then you can sort them from a mixed population. Another idea is that you could potentially have tethers embedded inside, which then you add the code post-assembly. So you can assemble your construct, and then if there is some kind of labeling code that you had actually in the media, then that would be kind of bound to your microtissue, and then kind of essentially track the conditions kind of after post hoc fabrication. So that's another element that we could do. And then you essentially need to enrich for phenotype later, and then decode what this exposure was after the fact. I just want to show one example of one way that we did this. If you guys have questions, you can ask me about it. But essentially what we were interested in was liver cancer and how it responded to chemotherapeutic, doxorubicin, and also in combination with treatments like an siRNA treatment. And so for this experiment, what we did was we encoded the siRNA treatment with the green signal illustrated here. So our control was green labeled. Our treatment with the siRNA was unlabeled. And then post-assembly, we encoded the concentration of our chemotherapeutic with what is essentially orthogonal to our fluorescence. It was a near-infrared signal, which you could detect after sorting based on a near-infrared scanner. And then, so essentially we had this combined experiment. So in the flow analysis, we're able to determine if a cell was treated with our siRNA or not. And then you can sort the cells below some viability threshold. And then we can do a post-analysis to then decode out the concentration of the doxorubicin that the microtissues experienced. So it's somewhat of a complicated experiment. I just wanted to highlight it here, essentially as a proof of concept of this multiplexing. So then, and as far as the kind of science, what we were able to identify was we saw this synergistic action. So if you had the treatment with the siRNA, then at the high concentration of doxorubicin, you had an increase in cell death that you don't really see under the other conditions. And so this is just two factors here, so you could very easily do this without this complicated multiplexing setup. But if you wanted to expand this to hundreds, thousands of drugs, then you could potentially encode these things in a batch system, sort them based on some phenotype, and then decode essentially post hoc what the relative contributions were. So we've been working on various ways to increase our multiplexing. One way is for improved fluorescent labeling. Another way is by essentially adding DNA barcodes. So in each microtissue, you could have a very specific DNA sequence, which then you can identify by PCR or sequencing, I think you guys talked about in this class. And so we did that by PCR here. So if you sort out based on some measure like viability, then you're able to decode what that DNA sequence was. And then, of course, we're interested in integrating this with other sensing modalities, many of which I think you've kind of talked about throughout this class, not just fluorescence or kind of standard molecular biology techniques, but other things as well. And finally, we're working more on this idea of injecting the microtissues into various host tissues. So in the experiment I showed there, they were encapsulated within this dialysis membrane, essentially like a bag, which allowed us to easily pull them back out. But what would be extremely useful is if you could actually inject them into a specific tissue, the spleen, the muscle, something like that, and then also potentially retrieve them from that tissue. And then you can do various in vivo experiments looking at various tissue sites and how they compare, or if you had an injury mouse model, something like that. And so I'm just going to kind of wrap up. So that's sort of an overview of a couple projects that we have going on in my group that integrate, and we're aiming to integrate with sensing technologies. And overall, our approach is trying to understand this microenvironment of how these complicated signals that cells integrate, stem cells, and how they become various components. And then there are a range of technologies that we and others that are kind of applying to deduce these processes. So I'll just wrap that up, and if you guys have any questions, I can take those. Thanks. Any last questions before? We asked a lot throughout.
I don't want to hog all the questions that I have on Phil. So we've learned about all these, the cost of DNA sequencing coming down.
Yeah.
And it looks like in your work, you're looking at different ways that cells start to undergo the differentiation process and then simulating things that might happen, inside a real embryo. Are people starting to apply sequencing to just all the initial stages of differentiation and now understanding what genes are active in each
yes, definitely. People are applying that, looking at the transcript level, but also epigenetic, like various chromosome modifications. So it's a big push, essentially, where you either in vivo in various domains, if you can extract those cells, or controlled in vitro experiments where you look at every stage of differentiation and essentially perform all the high throughput measurements that you can imagine, and then try it through various systems biology techniques, try to define the regulatory networks and essentially everything about the cells at all the stages. So definitely.
It must be a huge amount of sequencing, though. Is it inexpensive enough that people can do that, or people to do that? Or
yeah, I think it's getting there, and certainly for controlled experiments, I think it's well worth it. The one issue for the embryo experiments is getting enough source material to do that. There's so few cells. Increasing essentially the sensitivity of those types of assays is a big push, because the cell numbers are very limited.
So another quick question is there any people that think about using microtissues as a therapeutic, where you can inject microtissues into a damaged liver or damaged heart tissue?
Yeah, there are, and we've thought about that, and there are other people that have, where it might be a good delivery system where you could actually inject them into a tissue. So it's somewhat kind of in suspension to do that. Then also the flexibility of having different microtissues within one population, I think, could be very useful. So there are a handful of people, including us, that are interested in doing that.
Thanks very
All right,
much.
thanks a lot, guys.